A very good morning to one and all. Welcome to the day 3 of week 2 GIAN course. We'll start with the presentation. I request Dr. Hitesh Handa to start the lecture. So please.
Sure. Okay guys, good morning. It has been 8-9 days. My jet lag is still there. I slept for 3 hours yesterday. So if I say something weird, please forgive me. Okay. So today's class is actually most exciting for me because I'm going to discuss what my lab does. Last few classes were about materials, the issues with medical devices, fouling of medical devices, infection, clotting. Now my lab is working on a lot of things to try to mitigate the issues with the current medical devices. Okay. Also, please remind me that in the afternoon session, 15 minutes or 20 minutes before the session ends, we should stop talking about this and talk about tomorrow's exam. Okay? Tomorrow morning is your 2-hour exam. Okay. Let's start. Okay, I think it's a little bit perfect. So, conquering infections and thrombosis through biomaterial innovation. Before I start, I want to tell you guys a little bit about my background. Why am I doing what I'm doing now? A little bit about my lab. So I did my PhD under Dr. Gongzhao Mao in material science, surface chemistry, lot of atomic force microscopy, understanding coatings and films. Now, Dr. Mao is now the school head at University of Edinburgh, and I'm very grateful to her because when I went to her, I had just finished my undergrad from DCE, very minimum lab experience, and she was so patient with me and she taught me so much. Now, after I finished PhD under Dr. Mao, I went to medical device company. I thought if you are a professor, you have to do very basic mechanistic research, but I liked a little bit more practical research. So I thought if I go to industry, that's where I can do all that. If I become a faculty member, I have to do mechanistic research. Now, I worked for a medical device company for 2 years on developing nitric oxide releasing coatings for catheters and other implants. But while working there, I met with Dr. Bartlett and Dr. Meyerhoff. Dr. Bartlett was a co-founder of the company that I was working for, and Dr. Meyerhoff was scientific advisor to the company. They both were professors at the University of Michigan. Dr. Bartlett is a retired surgeon now, also known as father of ECMO. Dr. Meyerhoff is a leader. He also retired last year, but leader in nitric oxide releasing materials, chemist by training. So Dr. Meyerhoff was a chemist, Dr. Bartlett was a surgeon, and I had an engineering background. So after 2 years at the medical device company, they hired me at University of Michigan in Department of Surgery. And I think that was I'm happy that was a good decision that I made to go to work with them, because I learned a lot from them also, and they are the reason what I'm doing right now. While working with Dr. Bartlett, Dr. Meyerhoff, I learned a lot of animal models. I learned a lot of chemistry. I learned a lot about the clinical issues that surgeons are having with the patients. So working with them was a great decision for me. Now, a little bit about my lab. So, as you know, I'm at the University of Georgia. 2 years ago, they built this new building called STEM building. And this first picture is the building from outside. The second picture on top, that's my lab where the students sit. So that's the student office area, the cubicles. This is a conference room. So the building has a lot of such conference rooms because they want to really promote collaboration between the students. So there are a lot of rooms bigger than this one, smaller than this one, to encourage students to come and sit together, discuss projects, just to promote more collaborative discussions. And the bottom last picture is our kitchen outside our lab, where we interact sometimes or have lunch. And so really nice building, great atmosphere, very bright. So I'm lucky that we were able to get lab in this building. Now, okay. This is my lab. So we have almost 7,000 square feet lab space. There are a lot of support rooms like that one, the right picture on the top and bottom, both right pictures they are. So we have like a separate room for cell culture, for virus work, for bacteria work, for blood. One of these rooms we want to convert it to a surgery room. We just need to do lot of documentation for that, and then we will be able to do that. We have almost 25 fume hoods to work. So great place to work, and a great team. I'm very thankful for the students I have who are doing great work. So lab capabilities, we will talk more about the research, but mostly my lab has people with chemistry background, microbiology background, pharmacy background, and of course, lot of engineering background, chemical engineering, polymers, biomedical engineering, veterinarian, because we do a lot of animal studies. The goal is that we can synthesize new materials, design new surfaces, then we can do a lot of characterization. It can be AFM, SEM, FTIR, NMR, XPS, to make sure whatever chemistry you did or whatever new materials we are synthesizing, molecules, they are pure and they are what we think they are. Then we do nitric oxide releasing testing. Some of the tests we talk about, sterilization, leaching studies, all those we do. Then if those results look good, our surfaces look great, materials look great, we do a lot of in vitro studies, which are like protein adhesion studies, bacteria studies, blood studies, virus, if we are working with virus, cytotoxicity. So we work in a lot of those things. And if those results look good and the technology is very promising, we have animal models. Either we do it, we have done rabbit and mouse models in university. We can also do pig and sheep in the university, but I have collaborators who can do pig and sheep studies for us also. So translational research, by now you already know what is translational research. So when I was starting my lab 10 years ago at the university, I wanted to try to make it as translational as possible. Now, translational research is from benchtop to bedside. You're designing a surface, you are doing the testing, you're going to the animal study, and then you're trying to commercialize the material. And that's what we are doing. So we have development phase where we are making coatings, surfaces, catheters, wound dressings. We are doing all the characterizations, imaging, all that is looking great. Then we are doing hemocompatibility, antimicrobial, ELISA assays, toxicity studies. Those are looking great. Then we move to catheter studies, ECC models that I'm going to show today. If those are looking good and the work we feel is very novel, we file for patents, patent disclosures to the university, and we test those materials in animal studies. And whatever promising technologies are coming from the lab, I have a medical device startup company called Nitrix. We try to move those technologies and license the patents from the university in the company so we can try to commercialize the product. Okay. Now, we showed this picture before also. I got this image from Dr. James Breyer. Basically, we have so many different implants going on, right? So what I feel is, if we can make a truly antimicrobial surface or hemocompatible surface, there are unlimited applications for that material. Because a catheter coating that's really antimicrobial can also be used for other polymeric surfaces that are getting implanted. So one success for one application can actually be extended to so many different applications, can open a lot of avenues for other implants. Now, my lab works on a lot of different areas. Last night I was trying to, I'm going to show you some of the work we are doing, but I couldn't collect everything. But I'll be more than happy to discuss with you. Email me, I can send you papers if anything is exciting. On the website, you can see my website. If any paper looks good and you're interested, I can send you those papers. Now, mostly how it started was, I told you Dr. Bartlett is father of ECMO, or extracorporeal life support. And when a baby is on this ECMO, the baby has to be heavily heparinized. You can use ECLS even for adults also, but from Dr. Bartlett's perspective, he was doing it for babies, right? And you have to heparinize the patient. Heparin is making blood very thin. And even after you heparinize the patient, this is what's happening. All this black, still clotting is happening. A lot of clotting is happening. And people can be allergic to heparin also. So Dr. Bartlett, Dr. Meyerhoff, when I was working with them, the vision was, can we make a surface that we don't have to give heparin? Can we coat all the surfaces in an ECMO circuitry that we don't have to heparinize the patient? Or reduce heparin dose a lot so that we can prevent clotting of those medical devices. So that's how it all started for me. Now, this also you already know now, and maybe this is a good revision for you guys for tomorrow's test, that if you have any medical implant, proteins adsorb on the surface very rapidly, platelets get activated, adhere to those proteins, they call more platelets, they form cross-link, fibrinogen gets converted to fibrin, some red blood cells also get trapped in it, and now it leads to a clot formation, right? And all this is happening within seconds to hours, very, very fast. And you know this is clotting cascade. So this is like cascade of events are happening. Those factor 10 is getting activated, which is leading to another factor getting activated and just leading to clot formation. Okay. We saw this slide also, how hospital acquired infections are issues. So I'm not going to talk about this. And how biofilms form, right? I just wanted to still give you like all the background because I know some people are new and not attended the last class. So bacteria adheres to a surface, it starts multiplying. Approximately how much time for bacteria to multiply?
17 minutes.
It depends on different strains of bacteria, but yes, around 20-30 minutes it's multiplying, and it's also forming EPS, extra polymeric substance, which is protecting bacteria. And the problem is now you have to give 1,000 times the dose of antibiotics because they are not penetrating those biofilms. And bacteria is learning how to not die with that antibiotic. They are getting resistant to those antibiotics, right? So all these challenges are right now there. So our vision, Our human physiology is that our blood vessels release nitric oxide gas. Okay, and that's one of the mechanisms for our blood or platelets not to get activated in the body. Okay, this nitric oxide gas production from our blood vessels. Also, when we are sick, our macrophages, one of the mechanisms to fight infection is it releases nitric oxide. So nitric oxide is helping fight infection also in the body. And it's fighting clotting also in the body, prevents platelet activation. So our body is using this, it's our physiology to fight clotting and infection. Existing technologies have antibiotics, silver, heparin, lot of these different technologies trying to fight clotting and infection. First of all, you can either fight clotting or infection, nothing is fighting both, right? Antibiotics cannot fight clotting, heparin cannot fight infection, right? Plus, antibiotics are learning to not die from the bacteria, they are developing resistance. Antibiotics cannot penetrate biofilms, so even with antibiotics, infection is still happening. With heparin, clotting is still happening to some extent. And there is 5% population which is allergic to heparin, and it can cause HIT or heparin-induced thrombocytopenia in those patients, right? So our vision is, why not we make surfaces, now whatever it is, a catheter or a tubing or a vascular graft, whatever is getting implanted or going to see blood, can we make it nitric oxide releasing so that we can fight clotting and we can fight infection, and it's the same coating that can fight clotting and infection. Okay. Nitric oxide, so once it was considered a toxic gas, but in 1992 it was Molecule of the Year by Science. In 1998 these three gentlemen received a Nobel Prize for its signaling effect in cardiovascular systems. So it has been there. Lot of groups are trying to study it in different ways. Some are trying to understand the mechanism of it. Our group and some of my collaborators, I'm going to show some of them to you today. We are trying to design materials and make devices with those materials. This was just an interesting picture I saw online, that how our body, our blood vessels release nitric oxide gas, and that helps in our blood to properly circulate in the body. But as we grow older, the nitric oxide production keeps reducing, which can lead to thickening of arteries, inflammation, calcium buildup, and slowly as we grow older, the nitric oxide production is decreasing in our blood vessels, leading to more health and cardiovascular issues. Okay, so nitric oxide has lot of different roles in our body. Okay, it can promote wound healing, it can help with angiogenesis, regulates blood pressure. The main two things that we are going to discuss today is its ability to inhibit platelet activation and its ability to kill bacteria. Okay, it's a neurotransmitter also. It helps in, I told you, angiogenesis, that's growth of new blood vessels, so you can have a faster healing of the wounds. So a lot of different applications, but we are going to focus on platelets and bacteria. Now, nitric oxide is produced in our endothelial cells, and there was this paper in 1998 by Dr. Mark Vaughan from Texas Tech Lubbock. He did a computational modeling and showed that this is approximately the amount of nitric oxide that our endothelial cells are releasing. Okay. So 0.5 to 4, 10 to the power minus 10 moles per centimeter square per minute, or we can call it 0.5 to 4 flux units. So we understand, see, the study was done 20, 30 years ago, and it was a computational modeling type study. So we understand this number might not be accurate, but it gives us a really good idea of what to target at least. When we are designing materials, we want them to release nitric oxide. We release, this helps us. And we are not just relying on this number, because we will test that release with bacteria and blood, and we will test that release against toxicity also, just to make sure that the levels of nitric oxide we are producing are effective in killing bacteria, are effective in preventing clotting, but they are not so high that we are killing healthy cells. Right? Now, a question for you. You don't know anything about nitric oxide, and let's say you are starting the work in nitric oxide field in making catheters, let's say. What do you think are the challenges? I want to make a nitric oxide releasing catheter. What do you think are the challenges, can be the challenges with this field?
First would be toxicity. Too much of nitric oxide can turn into
So level of release, yes, okay. How not to have very high release?
So reactive nitrogen species in body causes a lot of inflammation and interferes with the body's immunity responses. So that could be another challenge.
Sure, so we have to really monitor the levels of release we have. Okay, what else?
How are we going to stick some bacteria, whatever the component is, which is going to release NO to the surface of the...
Okay, so if we have some molecule that releases nitric oxide, how are we going to stick it to my surface, right? Whatever surface it is. Okay, that's a good one. What else?
So as far primary, whatever, because nitric oxide when exposed to the environment, we have to care water. So ultimately in our body we are implanting something which is releasing nitric oxide. When it comes contact with water, it will turn into nitric. So monitor that also. Whether tide is actually working for us or nitric is actually working for us. That can be the thing.
Okay.
Five.
Okay, what else? Not couple of decades, we won't be able to do that, but yes, the longevity of release, like we don't know how much that is, but depending on the application, 1 week, 1 month, some number of days. Okay, what else? Forget nitric oxide. If you're designing any catheter, what do you think are the challenges? Sure, we want to make it anti-clotting, anti-infective, sure. But what can be other challenges? Yeah, sure. You know you cannot say biostable in exam tomorrow. Okay. Very good. how are the physical properties of the material going to get impacted, right? Because after you put nitric oxide donor in your polymer, are your strength is going to decrease by half, or right? That's not desirable, won't be accepted. What else? Okay. Actually, you are very right. So that's a good point. We did a study in that ECC model I showed you yesterday, where there is a tubing in the rabbit, and our coating was not good, and lot of nitric oxide was leaching in blood from the donor molecule that we were sticking in the polymer. It was not done properly, and that really dropped rabbit's blood pressure a lot, because we had like lot of bolus of nitric oxide release happening. Okay, what else? So do you think storage stability, right? Because when you're making a catheter, it's not going to be used tomorrow. It might be used after 6 months. Also, let's say they are making the catheters in Jaipur and it's so hot there, it's coming on trucks. In 2 or 3 days, it's reaching different places in India in that hot environment. So it's not just storage stability in freezer or refrigerator, but decent stability during transportation also, right? Okay, what else? Sterilizability, we talked yesterday. If we sterilize it and it's either chemicals or heat are destroying my surface, now that's a problem. And really big one, expenses. If it's very expensive, it's not going to become a practical solution, right? So when I started working in this field, these were some of the challenges. How can we get a long-term NO release? Can we get stable and manufacturing ability of this material? Can I store it and sterilize it? Nitric oxide donors are heat sensitive. Okay, so when they are subjected to high temperatures, for example, extrusion is done at a very high temperature. It will just destroy the material. If I do 120 degrees Celsius sterilization, it will kill the material. And another thing we will talk later, nitric oxide can kill bacteria, it can prevent clotting, but one of the limitation is nitric oxide releasing surfaces do not prevent protein adsorption. Proteins come and stick to the surface, and now nitric oxide is fighting platelets and bacteria, but that layer of protein is getting deposited on the surface. Bacteria is also sticking on the surface. Nitric oxide is killing it, but nitric oxide is not anti-fouling. It's not preventing stuff to stick on the surface. Okay, great. Okay, so then there are two types of, when we talk about nitric oxide releasing materials, there are two different ways to do it. One way is called nitric oxide releasing material, and I'll have a lot of examples I'll show you. And one is called nitric oxide generating materials. Okay, so the difference is, in nitric oxide releasing materials, we have nitric oxide donor molecules. We synthesize some molecule that has nitric oxide bound to it. We can put those molecules inside the polymers, and I'll show you some methods we do it. And now slowly that nitric oxide gets released. Okay, we will talk what is the trigger. That's one method of doing it, and that's called nitric oxide releasing materials. Now, nitric oxide generating materials, or no gen, is when we put a catalyst in the coating, which can be copper or selenium, for example, and our blood has these packets or RSNOs or S-nitrosothiols that carry nitric oxide. When this packet of nitric oxide carrying molecule comes in contact with this catalyst, it releases nitric oxide. So we are not relying on the coating that has nitric oxide donor in it. We are relying on the nitric oxide packets present in blood, and our catalyst is converting that packet to nitric oxide release. Okay, so nitric oxide releasing materials, nitric oxide generating materials. Okay, I think I have another slide where I have, in my 3-hour sleep, I thought I deleted this slide because I have more slides later, but I did not. So let's see how many other mistakes I have made. But this is not a mistake. This is one of the papers that talks about nitric oxide generating materials. There are more papers later, and the idea is same, but I wanted to put all the papers together. So, okay. Now let's talk actually about nitric oxide releasing materials first. Okay. Like you said, that there is a molecule that releases nitric oxide, right? And that's correct. There are different types of nitric oxide donors. Donor molecules that have nitric oxide bound to them, and they can release nitric oxide. So one such donor molecule is called diazonium dilates. Okay, or in short, we will call it DBHD. Okay. In our lab, we have nitric oxide analyzers. Basically, what this instrument does is it can very accurately and selectively measure nitric oxide release from the surfaces. So, for example, I make my catheter or film or wound dressing or whatever, put it at 37 degrees Celsius in a buffer. I can accurately measure how much nitric oxide this surface is releasing. Okay, and the number I can get is that 0.5 to 4, 10 to the power minus 10 moles per centimeter square per minute. We can get that same type of number with that instrument. So we know, am I releasing 1 flux or 2 flux or whatever flux I can release. So I can very accurately measure it. Now, diazonium dilates. So don't worry about this yellow or orange, whatever, don't worry about that. Basically, when I started working in this field 15 years ago, what was happening was, we have nitric oxide donor diazonium dilate. Let's say we put it in the polymer, we will talk about it. And in presence of buffer and 37 degrees Celsius temperature, it will break and release nitric oxide. Okay, but it also makes diamine species. So there is nitric oxide released and there is diamine byproduct formed. Now what happens is, when this diamine byproduct is formed, it makes the pH go up. So if I have a coating, let's say I have a polymer, polyurethane, I dissolve it in THF, a tetrahydrofuran or a solvent. Now I have a solution of polymer. Now I put desired amount of diazonium dilate in that solution, dissolve it, and now make it in a form of a film or a catheter. Now I know I have put 1 milligram of diazonium dilate in the polymer. Okay, but when I put this film in my nitric oxide analyzer with that 7.4 pH, 37 degrees Celsius, nitric oxide will release, but this byproduct of diamine species will form, and I only get this much nitric oxide release. So within 20 hours, so when I started working in this field, within 24 hours, the release will die. Okay, y-axis is nitric oxide release amount, and the x-axis is time. So, but I put diazonium dilate worth of, let's say, 5 days of release, and my release is dying in 24 hours. So this was the issue when I started working in the field. Okay. So one thing is, whatever I'm showing today, it's all teamwork. You know, my mentors help, my collaborators help, my students help, my postdocs help. So it's nothing that my work only, it's like we all work together. Somebody is a chemist, say they gave a really good idea. Somebody is a microbiologist, they gave a great idea. So that's how science moves forward. Okay, so we talked about this. Now, during my PhD, I published, so this is important for all of you. A lot of time we think, oh, my PhD is in this area, I'm never going to use this again in my life. I don't need to pay attention. You never know. So in my PhD, I worked with this professor in pharmacy, Dr. GIAN on some extra projects other than my PhD projects. He was a big drug delivery guy with PLGA. I didn't know what is PLGA at that time. So I worked with him on PLGA drug delivery type applications. So I remembered that we talked how PLGA can break to form lactic acid and glycolic acid, and PLGA is FDA approved. It's not toxic, right? You can have PLGA that degrades very fast. You can have PLGA that degrades very slow. And it's very soluble in a lot of solvents. So, and this was like 3-4 years ago I did this work, right? So then we discussed and thought, why don't we use and put PLGA in our coating? Okay, so now I have a catheter material. I put my nitric oxide donor in it. What if I put little bit PLGA in it? See, I know for a catheter, I don't want a biodegradable material, right? But if let's say 5% of my coating is PLGA, 95% is polyurethane for strength and whatever, right? What will happen is, what was our assumption was, our hypothesis, that when this reaction will happen, when diazonium dilate, my nitric oxide donor will break and form diapine. At the same time, PLGA will also break and make acids. And don't worry, I know you're thinking, oh my goodness, your acid in the blood will change. We are talking at like very minute amounts, right? Only acting inside the coating. So at the same time, you're making diamine, but at the same time, you're forming these acids, and these acids and diamines can neutralize each other, and the coating pH can remain neutral or slightly acidic. So this system can keep releasing nitric oxide. Okay. And see, I'm showing you like 2 years of work, but thinking is one thing, making it happen is another thing, and then you are not finding the right PLGA is another thing. So there is a lot of optimization. Okay, so what we did is we designed our coating, and let's say we use polyurethane. You know, we tried a lot of different polymers, PVC, silicon rubber, polyurethane, but let's say polyurethane. So we made a polyurethane film. We added polyurethane, diazonium dilate, PLGA in it. But then we put a top coat of only polyurethane. Any reason why we did a top coat of that polyurethane? And see, I ask these questions to force you to think. You know, I'm not expecting you guys know this, but I'm just trying to forcing you to think a little bit. Active layer is inside, and then outside top coat is just the polymer. So my top coat is not PLGA. My top coat is only polyurethane. My top coat is not degrading. It's not PLGA. PLGA is inside. The top outside coat is just polyurethane. Okay, okay. And what else? Yes, that's fine. What else? That's good. And also remember, let's say you have a catheter. If my outside PLGA is there and it starts degrading, I'm making tiny structures on the surface, right? It's protected now inside. Even if it's little bit breaking, 5%, outside is not seeing anything of that little bit holes in the surface or not holes or whatever, right? Another reason is we want to reduce leaching of my NO donor. See, I want my NO donor to keep releasing nitric oxide in presence of like acidic environment, 37 degrees, moisture, but I don't want my whole NO donor molecule to come out. See, if lot of molecule comes out, it's going to be toxic, it's not going to last long, it'll release a lot at the same time. So it's protecting it inside and not letting it leach out. Also, you are smoothing the surface from outside, little bit we talked, and you are neutralizing the surface charge because of diazonium dilate. Okay. So when we were designing these surfaces, we thought a lot of things. So if you were designing this surface, what all would you think? This is your first PhD project. You are designing a coating with PLGA polymer. What all will you think about when you're doing this? A lot of time in you know, will meet with you and give you like a big idea. You can break and acidic environment. You can have diazonium dilate. Okay, this is your first PhD project. Thinking and designing a project. What all do you think about it? What thought will come to you? Like, okay, now you are going back to your lab and now you have to start doing something. What all would you think? Okay. Okay. And remember, we are not releasing DBHD. That's an important thing to remember. I know you didn't mean that, but I'm just trying to again and again say we never want NO donor to come out. We just want the gaseous nitric oxide to come out. And it's very easy for nitric oxide to just get released and come out. Okay. Anybody else? So NO it's a small gaseous molecule. It can penetrate through all these polymers. Okay. I also forgot to mention nitric oxide has another benefit. It can penetrate biofilms. Because it's a small gaseous molecule, antibiotics have difficulty penetrating biofilms. Nitric oxide can penetrate biofilms. Okay, sorry, that was off topic. What else? Very good. Yes. Yes. So this is what we did, right? This is why it took us so much time. Thickness of the coating. But also remember, in the real life catheter example, we cannot play too much with the thickness, right? Because we need to have certain inner diameter, we need to have certain outer diameter, and we have very little room to play with the thickness. But you are right. Can we make active layer very thick, top layer very thin? Compared to each other, how much we can play. So let's say, I'm just saying example, let's say 800 micron is my total thickness allowed. Should it be 400, 400, 200, 600, 600, 200? So we played with that a lot. Okay. What else? So we try to find a, there could be. Very good. So we want to think what should be our base polymer be? Like, should we use PVC? Should we use polyurethane? If polyurethane, what type of polyurethane? So different polymers have different water uptake. And in this type of application, water uptake is very important. Why? If it's hydrophobic, that's an issue because water is not penetrating inside. Until water will go inside, this coating won't start breaking or releasing, not breaking. So we want it to be little hydrophilic. But if it's too hydrophilic, that's a problem. All the NO donor might leach out of the coating, and that's a problem. So we want a balance of how much water uptake is there or how little water uptake is there. So we have to decide the polymer. What else? It's all there. Huh? Sure. But what else? It's there. We are not even worried about testing. I'm just thinking you're going to lab and you want to start making your coatings. What all are you thinking? Smoothness, we know if we'll do the top coat, we have smooth surface. Do you think you have to think about how much Dysanium dilate to add? Yes, quantity of each of the quantity of PLGA and quantity of Dysanium dilate and polymer. I mean, polymer will be maximum. Yes. Because that's giving the structure and strength to the material. Other two things are, see, PLGA has to be very less because we don't want it to start dissolving and your catheter is dissolving now, right? Dysanium dilates, if it's like a powder that's nitric oxide releasing. If you add too much Dysanium dilate in a catheter material, it might start becoming brittle. So now we had to play with what's the ratio of all these things. Okay. Then another thing was which type of PLGA. Do we want a very fast degrading PLGA or do we want a very slow degrading PLGA? Okay, so these were some of our drawing board things to do. Okay, and see, it sounds okay, but if you start doing it and start making coatings and then start testing, you want to test NO release also, you want to look at the strength also, you want to look at the leaching also. It can become a lot to then narrow it down that what's the right material. Okay.
So, do you use any kind of binding material also to get all these DHT, PLGA to stick to a base layer?
No, so it's just suspended in that coating for this work.
So, in order, if we are, let's say, if we are designing catheter that would go in the market, so will there a binding material required?
So yes and no. We can just suspend it We can covalently bind it to the polymer chains. We have tried all these different things. I'll show you. So there is, yes, if you don't bind it, you want to be sure it's not leaching out. If you covalently bind it, now it's a little bit safe because it's not going to just leach out. It's bound tightly, right?
I just feel that there is polymer, like the coating of all of these materials that
You can do that also. Or you can make a solution and mix everything up and then cast a film. Or you can do layer by layer coating. So for example, this is off topic. When we make catheters, what we do is we take a solution, whatever that solution is with NO donor or whatever, and we take a metal mandrel and we dip coat it. Let's say one dip coat every minute. So I dip coat it, wait 1 minute, dip coat it again, wait 1 minute, do 30 dip coats, 40 dip coats, 50 dip coats. Let everything dry. Basically, dip coating has put lot of layers of that polymer on that mandrel. Remove the mandrel, you have a tubing or a catheter ready. You can have, first few dip coats of your control material, of your base polymer. Then you can do some dip coats of your active coating. Then you can again do some dip coats of your control material. Then in that mandrel, your inside first coat was just base material, then dip coats of your active material, then dip coat of your base material. So when you remove the mandrel, inside and outside of the catheter is base polymer, sandwiching your active material in the middle. Right?
so like picture we can see it's just printed in the same thing. Yeah, multiple that prolonged
We have tried a lot of things. So multiple sandwiches you mean is one coat, top coat, another coat, right? See, that's a good idea, but thickness is a limitation. Now, you can add another coating also if the polymers are very different from each other.
It's the same polymer. Uniformity would be there in the polymer. The
Okay.
active coat will also remain the same. It's just multiple elements.
Oh yeah,
And
yeah,
thinner than this.
yeah,
Not this thin.
yeah. Of course, you can play with all that.
So like micro of that polymer, and we can have like 150 or 200, and we can do
So you are saying layer by layer. You have NO release layer, control layer, NO release layer, control layer. Yeah,
diversified
yeah,
things.
yeah. See,
But so if we're doing this like NO layer and then when the NO is released,
It can release its gas.
it can come out.
Yeah.
Yes, it can come out. And see,
no option.
you are on the right path. This is how people in my group think. Like, this is the way we think and design new materials.
We always consider the limitation and restriction first.
Yes.
And then think around it.
Yeah, and then thinking out of the box, because so many people are doing so many things, right? Some are our collaborators, some are our other scientists who we don't work with. Everybody's trying all this, and the goal is same: how to make a stable material, control release, longer term release. Okay. Then, forget borate. I don't want to tell you the borate story. That will take some time. So forget borate. Forget it. Now, this is an example showing when we did just a polymer with dizanium dilate with a top coat of polymer, this green is what happens with the NO release. In 3 days, NO release is dead because my coating is becoming so basic that release is stopping. Okay. But then when I add PLGA in this coating, the red one, I can get release for 14 days. So the idea we had, hypothesis was right. that you can add little bit PLGA, it's keeping the environment little bit acidic, little bit neutral, and you keep releasing nitric oxide. Okay, then, yes.
So why is this borate and control missing in 710?
Oh, it didn't last.
Okay, it's exhausted.
It exhausted, yes, yes.
And so control here is?
Control is your polymer, same polymer, has diazonium dilate in it, DBHT, and a top coat of polymer. Your active is everything same, but with PLGA.
PLGA is missing.
Yes, yes. Control and borate have no PLGA. Don't even worry about borate. But control has no PLGA, and your PLGA is the only additional thing you have.
One more question. Like few slides back, you showed that the blue and yellow lines dotted graph. Now,
Yeah.
some borate, tetraphenyl
Yeah.
borate additive, but it was toxic. It was extending the release, it was toxic. How
Yeah.
do we deal with that?
That's why we cannot use it.
Also,
That's why I didn't even want to go in that story. So that was being used when I started working in the field a little bit,
Okay, just to explain it.
but I didn't want to go in that direction. So I just, yeah. Okay, then what we did is, we got different types of PLGA. Okay, a very fast degrading PLGA and very slow degrading PLGA. 1 to 2 week degradation and 1 to 2 month degradation. And when we did NO release, see, this is interesting now. You have to pay a little bit attention. Black is 1 to 2 week degradation PLGA. Okay, black. So that means it's degrading very fast. That means it's creating very acidic environment in the coating. That's why the release is so fast on day 1. But it's dying by day 10 because you released all your diazonium dilates very fast, NO release very fast, and it died quickly. But when we did 1 to 2 month PLGA, we had a more consistent release for a little bit longer period of time. I don't remember now. Maybe we tried 1 to 2 year PLGA also, and maybe that was too slow and we couldn't see the effect of it.
One question.
Yes.
In the black one, since it's usually acid very fast, what will cause, since the polymer is a kind of metal only, not a metal, but would it cause a acidic corrosion or something? It's too fast?
Not acidic corrosion, but can be toxic. Like the levels of nitric oxide release are very high, more than 10 times what that computational modeling number showed. It might have been. See, the thing is how science moves. I published this paper 10, 12 years ago when I was working with my PhD mentor, Dr. Bartlett. We were not doing toxicity studies at that time. We didn't have the facility to do toxicity studies, so we never tested this material for toxicity. But it could have been toxic, you know.
So what's the difference? 1 to weak PLGA, 1 to month, or 1 to PLGA?
So PLGA is polylactic co-glycolic acid. The ratio of lactic to glycolic acid can make it a very fast degrading or very slow degrading PLGA.
On the third day in that graph, the PLGA, second PLGA, 1 to 2 months, it rises. The drug release. So it is due to that the degradation is happening, or
So first of all, this much little bit up and down, it's okay, you know, but next slide will show you what's happening. Okay, let's pay attention to this slide. This was very interesting also. So there is this dye called bromothymol blue. What this dye does is, if you add this dye to a very acidic solution, the solution will look yellow. If you add it to a basic solution, your solution will turn blue. Okay. So what we did, okay, we think this is what's happening, right? That PLGA is breaking, acidic environment is creating, diazonium dilate is breaking, basic environment is forming. This is what we think. We wanted to show it more qualitatively. Okay. So now what we did, pay a little attention again. We added little bit of bromothymol blue dye in our coatings. Okay, let's see control, which has no PLGA. You start with a bluish, little yellow tint coating. It releases a little bit nitric oxide for a day or two, makes it more basic. Your blue color becomes darker in the coating. When we had 1 to 2 week PLGA, your coating is yellow because your PLGA is breaking so fast that it's making very acidic environment inside the coating. But that's releasing nitric oxide, making some diamine species, making a bluish, pushing it to bluish color because it's fighting to make it basic environment. That's why it's landing green color, because it's somewhere in between yellow and blue. But by the time it's day 7, all the nitric oxide is gone. And now PLGA won the battle. PLGA is making it acidic. There is no nitric oxide donor left to make it basic anymore. That's why it turned yellow, that it's done. And that what it matches here also, that by the time it's day 7, day 10, your NO release is dead. But when we had this 1 to 2 month PLGA, the coating remained greenish for a very long period of time, because there was a very nice balance between NO release and PLGA breaking. And see, a really good application of this is if you have a catheter in a hospital setting and you tell the doctor, if this catheter turns yellow color, cap or something, that means NO release has died. That means all the NO donor is gone and you just have a yellow color now. Okay. So this was the story of diazonium dilate molecule, nitric oxide molecule. Okay. But later, we started working a lot with SNAP or S-nitroso N-acetyl penicillamine. This is another NO donor molecule. Okay, diazonium dilate is one type, which is a little bit more complicated to synthesize. You need a high pressure system in the lab and everything. We have it, but this one is a little bit more convenient to work with. Now, SNAP is also synthesized from NAP or N-acetyl penicillamine, which is actually FDA approved for heavy metal poisoning. So our idea is also, if we are using something that's FDA approved and synthesizing SNAP, maybe going and getting a FDA approval might be a little bit easy to go that route. Okay. Now, unlike diazonium dilates, we don't worry about acidic or basic environment here. Heat, light, metal ions, moisture, those are the triggers for nitric oxide release. Okay. But then, like heat is a trigger also, but that means it's heat sensitive also. Even diazonium dilate is heat sensitive also. And that's important, right? That's why at a body temperature, it starts breaking and releasing nitric oxide. So we want it to be a little bit heat sensitive also, or break with heat or light or metal ions. Okay, so with this work, when we started working, we thought we again wanted to go to the same question, which polymer to use. So we tried a lot of different polymers with different water uptake. Silicon rubber, carboxyl is a copolymer of polyurethane and silicon rubber. E2AS is a copolymer of polyurethane and silicon rubber. SP60D60 and SG80A are polyurethanes, just two different types of polyurethanes. Okay. Now, but remember one thing, the top 3 polymers have very low water uptake, 1%-ish. And the bottom 2 have very high water uptake, 6% and 60% water uptake. So what we did first study was, we made this solution of different polymers, put SNAP in it. SNAP is an anode donor. We don't need PLGA and all that now, so forget PLGA now. And we casted films. Put them in buffer for 24 hours. And what we saw with UV vis is that SG80A SP60D60, all the anode donor leached out within 24 hours. Very fast. In 4 to 8 hours, most of the anode donor was leached out. So those are not good candidates for us. I don't want my anode donor to leach out. But the other three were really good. They almost kept all the anode donor inside. Because the water uptake was so high, water was entering inside and bringing all the anode donor out. Okay. This is just an example showing how we can switch on the light and increase the NO. We can switch off the light and reduce the nitric oxide level. A good example of this can be, for example, wound healing. You know, you have a dressing. If you feel every day, oh, infection can happen, your nitric oxide levels are only 0.5 or 1, shine light for few minutes or 1 hour or so that you can have higher release of NO happening and you can take care of infection and then you can switch it off. Okay. Another idea, we have published a paper also, but I don't think so I have a slide on it. It's a little bit off. We have not reached there yet, but I will forget if I won't tell you now. So we have also done make a nitric oxide releasing SNAP catheter that can be implanted, but have fiber optic cable that you can insert inside this catheter. Because fiber optic will release light, that light will trigger more nitric oxide release. So you can have that fiber optic thing going on for few minutes or few hours, or you can even have like a short fiber optic, because a lot of time ports that you are using can get infected. You can have a port connected with the fiber optic. After you take the blood out or infuse something, put this cap on with fiber optic so that it can release more nitric oxide and take care of infection if there was any infection, higher levels. With this SNAP method, we were able to prolong the release to 20 days. See, first when we started, we were at 1 or 2 days of nitric oxide release. Dizanium Day-8 helped us to reach 14 days of nitric oxide release. SNAP helped us to reach 20 days of nitric oxide release. Okay.
So if the catheter, I mean, it kept here like.
Yes, yes. So a lot of time. So yes. So these materials, they can be light sensitive also. So a lot of time we just store them in dark places. But that's easy to take care of. You can have a packaging material that's dark, like opaque.
So no, I mean, I'm even so I insert a catheter.
But see, insert is, it's inside, right? So that light is not reaching inside. Right?
So when you were testing this light sensitivity with optical fibers, the problem that I see is that is only at a specific point. Or something.
So we can, right? You know, if you think the port might get infected, we can have a short fiber just there. If you think it's longer, we can just put a little longer fiber inside. You know, we have lock solution. So instead of a lock solution, you have this lock fiber optic cable, battery operated, that you can have. Or you can only have it for few minutes or few hours. Immediately after a procedure, have that cap for 1 hour, have the light on, and then after 1 hour, you can switch off the light, you're fine. If any bacteria was introduced during the procedure, you took care of it.
So,
Yes.
what method do you use to keep check on the time for which the light is being released by that optical?
So, with the nitric oxide analyzer we can do all the in vitro study, right? That how much time, what intensity light we have on, what are the levels of nitric oxide release.
If the catheter have shelf life. So, I mean, if it is releasing nitric oxide and suppose it's, as you said, the time is for 20 days and it is packed and it is kept like that, if I've not used it, then also the NO will deplete.
No. So 20 days is testing at 37 degrees in buffer. If it's coming in 2 slides or 3 slides, I think. When we are storing it in refrigerator or freezer or on the shelf, it's not releasing, maybe it's releasing less than 5% nitric oxide because it's not sitting at 37 degrees in moisture all that time.
We have to take care of the conditions it is kept in so that it does not start releasing.
Yes. Of course, if you put that catheter in liquid, then nobody will use that catheter anyways. But if it's kept at room temperature or in fridge or in freezer, in refrigerator, freezer, it won't even release anything. At room temperature, dry conditions, it might release 5% of it. It's coming in 2-3 slides, that data. Okay, so Marcus was my first PhD student and he did some work where he tested a lot of these things from commercial side of things, and I'll show you a little bit of this from Marcus's studies or other studies. But the point was, how are the surface properties? How are the mechanical strength, sterilization effects, NO release? And I have put the article, if you're really interested in a little bit of that, there is an article, you can read all that, but he did a lot of studies like that. Storage stability, so a lot of those things. Okay, this is another good slide. Actually, this slide is 10 years of our work. I've tried to show it in this way. In 2009, when I started working in this field, we used to have 3 days of nitric oxide release. I showed you already, right, without PLGA. Then we published a paper with PLGA1 and we reached 14 days of nitric oxide release in 2012. In 2015, we had 28 days of nitric oxide release with some snap things. But then in 2018, we published a paper where we had a 4-month of NO release. So to give you an idea, if you show it to somebody, it looks like one graph, but this is a journey of 10 years to go from 3 days to 125 days of nitric oxide release.
So what would you employ?
So this material was silicon rubber, I think, PDMS.
So that the
What do you mean?
the material, like on the
Oh.
14 days PLG, 8 days.
Oh, okay. So let me go back a little bit. So this graph is a little bit misleading in a way. This was all silicon rubber data. I have just shown you, this is all one material of 125 days. I'm just showing you that in our goal to achieve longer term NO release, different materials we tried, different combinations we tried. In 2009, we reached this goal. 2012, we reached this goal. Something was done with diazonium dialate, something was done with SNAP, something was done with PLGA added. but then we were able to achieve 125 days of release. Okay, so this is your question, Neha. We did a shelf life storage study. Actually, we even have a data of 6 months now. I just didn't put that data here, but we have 6 months data that how in freezer and dark and room temperature and 37 degrees Celsius, how much NO we are losing. But it's all in dry conditions. So we lose less than 15% of NO. And actually, with the newer materials we have, the 6 month data, we are losing only less than 10% of NO in storage while shelf life. We did sterilization study. Actually, this is Marcus's paper also. He has some of this data. That we tried different sterilization methods, autoclaving to UV to ethylene oxide to plasma. Autoclaving was bad. Everything else was fine. We were able to have more than 90% NO release capabilities.
So what exactly happened at 50 degrees Celsius in that graph?
In what happened at 37 degrees?
50 degrees Celsius.
50 degree. Oh, I think maybe we didn't show it because maybe it dropped more. So we didn't do. Also, I think our thought was also now, I think we only did 1 month of 50 degree, thinking if we are shipping something and now it's stuck and sitting in a truck in Delhi for 1 week, then temperature in that truck might reach in 40 degrees or 45 degrees. So that's why we only did 1 month. It better not be sitting in a truck for 6 months. If the truck is delayed or something, that's why we only did for a month of that very high temperature sitting.
Cannot use after.
Yeah.
No, but actually my question was what happened with that
Why it? What happened means after.
like for 37 degrees Celsius it is prolonged for 4 months, but in 1 month the 50 degrees Celsius it dropped so much. Why?
Yeah, because temperature was high, right? We are losing more nitric oxide.
Oh.
And also, we are not going to store those catheters at 50 degrees. I mean, that will be ridiculous to store it in outside in sunlight.
Within a month, at 50 was so penny that it was not.
Also, this study we did only to prove that if by chance a catheter is getting, see, catheter should be sitting in a room temperature on a shelf or let's say in refrigerator. But if you are shipping it, it better reach wherever you are sending it within a week or two. Worst case, it's a month. You might lose 15% of NO, but then you can keep it in the refrigerator and you will maintain that level now. Right? It should not just be sitting at 50 degrees for 3 months. We should not. And if that's the condition, then maybe this is not the best technology.
Suppose if it was 1 month,
Yeah.
it lost some amount of NO. So it will also reduce the shelf life of the catheter, as in that after 1 month, it can only be used for a certain period of time. And
So,
because they have already lost, suppose 50% of NO, and there's only 50% left, and
yeah,
we can only use it for some period.
True. Also, see, remember, if we market this type of catheter, we are not going to say 120 days. There'll always be some buffer. See, if a medicine expires in December, you might actually label September that, hey, medicine, that like everybody's keeping that extra buffer that you're not saying absolutely, oh, my catheter is 120 days. Maybe we will only market it 60 days. So you are already considering those things. And I think if we go forward in that direction, maybe FDA will ask those type of questions. That, okay, if you lose 15% of NO release, and I'm almost sure we already have that data also. Somebody must have done it. If you have lost 15%, Now what's your NO release trend? Are you releasing less every day but still lasting 120 days? Or now you're lasting only 100 days and not going 120 days? So all those things will have to be discussed, right?
So, why do you think in all the four situations that we're keeping in, on the 15th or the 20th day, there is a sharp dip, and then throughout the duration, there is not a like significant change drop in the material?
Okay, that's a very good question. So we found out that the SNAP NO releasing materials, they form crystals inside a polymer. Okay, but some of the SNAP is not crystallized, little bit. So what is not crystallized, that can come out fast. But what's crystallized now, that will release very slowly. When moisture will go inside, it will try to little bit dissolve a crystal and bring little bit NO out. So you saw that little sharp down because of that uncrystallized snap that's sitting inside the polymer that came out faster. Mostly for any NO release polymers when we make, we see that with NO release also. Lot of times we see little bit higher NO release on day 1 or 2, and then it starts going little down because of that uncrystallized snap that's just sitting around there.
It's actually good that it's very crystallized.
Yes.
Preserving for longer.
So that's why we were very happy when we got 20 days of NO release. That's stabilizing snap inside the polymer. Otherwise, the NO donor can just run out, but it's giving it a more. So since you started that conversation, we also found out if we put up to 10 weight% snap, or sorry, up to 5 weight% snap, it does not crystallize. But after that, some amount, 4 or 5 or 6 weight% snap by weight, so 95% polymer or 90% polymer, that's the quantity you need for the crystals to start forming. Okay.
So we are making sure that we use quantity in that range.
Yes, but we also showed if you add 15 weight% snap or 20 weight% snap, it will happen,
But less of it.
but your material becomes less strong and it starts becoming a little bit brittle. You are compromising on the strength of the material by adding too much NO donor. Otherwise, you can always say, hey, let's add 50% snap. That's great. We can have more NO release, but no, the strength of your material will go down really tremendously. Okay, so next interesting thing is, oh my God, today I think I need to drink more water. Thirsty. So next interesting thing was, so Professor Brisbois is working, I'm working with her for last 10, 12 years. She's a collaborator. I told you these NO donor molecules are very, if I sit here, it's okay, right? I can sit? Okay, great. Oh my goodness. Oh. Okay. So, NO donor molecules are very heat sensitive. But when we are, I mean, I can stand for another.
I mean, you can sit here.
Oh, okay. This is better. Okay. Great. Okay. So, NO donor molecules are very heat sensitive. But how do we extrude now? To make catheters, tubings, there is extruder, which is like pellets of polymers are dissolving. You are putting NO donor in it at high temperature and it's forming into tubings or whatever. But we saw autoclave. Autoclave basically killed all the NO release because of high temperature. So this paper was very good. in Acta Biomaterialia. What we showed here was that if we swell polymer tubings, so what we do is we have a solvent with NO donor dissolve in it. We take existing catheters or existing tubings, swell it in that solution for, let's say, 24 hours. Idea is that solvent will take the NO donor molecule into the walls of the catheters, take the catheter or tubing out, dry it overnight or 2 days or whatever with vacuum to make sure you get all the solvent out. And now all the solvent is gone, all the NO donor is inside the wall of the tubing, and now you have a NO releasing material ready without extruding, without making a film or whatever, and it's very uniformly distributed inside the wall of the tubing, and you have a NO releasing surface ready. And
I had this doubt that what if instead of we can just make
yes. So now you can pick a existing tubing, existing catheter and make it NO releasing. This paper, like, I mean, you can look at it. I don't even remember. Oh, we did even rabbit study with it. I just wanted to tell you the idea. We did a lot of studies to make sure NO release was good. The surface in the SEM looks fine. It's not that surface is becoming rougher now. We tested it in the animal model. So we did all that with this. Okay. Then another idea. See, I have a lot of slides like this now. Okay. One slide, one paper, basically. So I'm just giving you a basic idea. If you're really interested about the idea, you can go to that paper. Okay. I ran out of time when I was making the slides because I wanted to make like this. I have a lot of slides like this, but the paper information is there, the manuscript, so you can find it. So other idea was, we always have this reservoir of nitric oxide donor in the polymer. If it dies, my nitric oxide release will die, right? So we thought, why not we use dual lumen catheters? So there is a wall in between a catheter. It has two sides. Fill one of the side of the catheter with nitric oxide donor with some hydrogel, like PEG or whatever, and seal it. Now the idea is NO release that's happening from that lumen is protecting, NO release is going in all directions. It's protecting the other lumen that you are using to take the blood out or infuse something. Another beauty of this is every few days, let's say NO release from this lumen is 14 days. After 14 days, you can put a needle in this lumen, take this hydrogel out and refill it. Now you don't not worried about, oh, my NO donor will run out. Okay, so this was another paper. This is just an idea, like we wanted to just show it in a, instead of showing it in lumen, we made catheters, we filled NO donor in it, and we showed that how when we just fill snap powder in that lumen, we get a very low NO release. Okay, but if we fill PEG, which is a hydrogel with NO donor, we get a really good NO release because it's a moisture environment and hydrogel is helping NO to break and release. And here we are also seeing nitric oxide donor snap is green in color, that synthesized molecule. Over 14 days, how that green color is going away. So by the time it's almost 14 days, all my NO donor is gone, and I can refill the new one. Actually, I'm so sorry, I've said this was NO release. This was not, I just saw. This is water uptake. So of course, if you have more water uptake, you can release more nitric oxide. So with only snap powder, you have water uptake is very low in that lumen. But if you have a PEG in there, you are helping with more water uptake, which can help in more nitric oxide release. Okay. We already talked about this. Antimicrobial testing, bioreactor, you can homogenize a sample and then you can plate it. But this is just one study with one of these materials. This is not manuscript under preparation. It must be already published. When we make nitric oxide releasing materials and do a 7-day bioreactor study and do live staining, live dead staining, lot of green bacteria, live bacteria, and mostly dead bacteria with NO release surface. Okay. Now we have next chapter of this thing. Let me do one thing. This is like a whole story. Let me skip some slides and talk about some different things, because this I want to do it in continuation after lunch. Okay. So let me actually go to, and I'll tell you guys which slide number. So let's talk about a little bit this one. Okay. So we did this work in around, 86. Yeah. We did this work in around 2013, 2014. Acetobacter baumannii is a big threat, and it's a very drug resistant bacteria for wound infections, multi-drug resistant bacteria. So what we did, the study was, we used Dizanium dilate material with PLGA. We made a wound dressing with that polyurethane, PLGA, Dizanium dilate. And what we did, we took a mouse model, we gave a little burn wound at some 50 degrees Celsius water or something, and then we infected that wound of the mouse with Acetobacter baumannii. And then for 24 hours, we applied our wound dressing, and for 24 hours, we applied a control wound dressing. And then after 24 hours, we homogenized that tissue and that dressing, and we looked at the infection. So we found 4 log reduction. So see, I hope you remember we talked about it. 1 log reduction is 90%, 2 log reduction is 99%. So 4 log reduction is 99.99% bacterial infection was reduced. Another, and I cannot tell you the details a little bit here, filing for patent and all that. We made some wound dressings that are loaded with drugs, whatever drug we want, NO release or clotting agents or whatever, but the dressing is completely biodegradable. Because a lot of time what happens when somebody has a burn wound or whatever very bad wounds, if you are coming and changing dressing every day, whatever cells had started growing, you are removing it again to put a new dressing. So the idea is you don't need to remove anything. You just put a new dressing every other day or every day, and it's releasing the drug, but the dressing is also dissolving while releasing everything. Another property of NO is GSNO is another type of NO donor. So don't worry about it. SNAP, GSNO, similar family little bit. We also, NO helps in cell proliferation. So if there is a wound, so here we maybe you cannot see it very clearly. We had cells. We put a cut so that there is a place where there are no cells now. We removed it. And with the dressing, we showed how the cells grow closer to each other faster. So they are helping with the growth of these cells and faster healing of wounds. Okay. Yes.
This
Ooh, yeah, this one.
one. Which
Last picture.
this one?
Oh, this one. Yeah, okay.
Control, you see that there's this rectangular red box and then yellow inside linings are there. Is that place where the wound was made?
Yes, yes, yes.
There are no cells visible there.
Yeah, you know, this picture is not great, but yeah, there were cells. We removed the cells. So there is also now, instead of doing scratch, so you call it scratch test. Now you also have these, I don't know what to call it, these inserts that you can have the insert. So when cells are grown, they are not grown in that insert area. You remove the insert and now you see how fast you are healing that area, how it's coming closer to each other. You know?
Later, after 24 hours, we what's there?
Yes.
So there are cell growth over there.
Yes. Okay, let me show you something else because I do not want to start that chapter. Okay. Okay, let's talk about this a little bit. So Katie did her master's with me. So she worked on endotracheal tubes. So we bought endotracheal tubes and she made them NO-releasing. And then we did a lot of studies. You can go to this article and read more. But we did bacteria studies. We did leaching of SNAP. We did tensile testing of the because we don't want the endotracheal tubes to become too brittle and less strong. So we did a lot of those studies to show we can make endotracheal tubes more antimicrobial. Okay. See, these were the slides I was making, and I was half asleep. So there is no good order to this. And there doesn't need to be any order to this, honestly. They are all independent papers. But since our break is coming soon, I don't want to start a chapter that will not be. Let me show you now. Okay, this is a good one. So slide number 97. Okay. So Laurie did her PhD with me and graduated, I think, 2 years ago. So this study was, I really like this study because a lot of times, most papers we have and others have in the field, we always infect the medical device with 10 to the power 7 CFU bacteria per ml, approximately. You know, that's a very high load of bacteria. And a lot of time we show, oh, my nitric oxide or whatever materials people are testing, we have 1 log reduction, 2 log reduction. So we thought, why don't we try different bacteria infection rates? You know, 10 to the power 3, 10 to the power 5, 10 to the power 7. And it was very interesting. We basically showed, read this paper if you're interested. We basically showed that if your infection is 10 to the power 3 CFU per ml, nitric oxide can take care of the whole infection. But when the infection is 10 to the power 7, it's so overwhelmed with bacteria, it's fighting, but it can only kill 90% or 95% or 99% because there is so much bacteria and there is not enough nitric oxide to take care of it. But in real life, most of the time when somebody gets infected, they are not going to get infected with that big bolus of bacteria. Mostly patients will get infected with 100,000, 10,000 bacteria at a time. And if we have this nitric oxide releasing devices, we are taking care of that infection very fast. You know, so I like that paper because we wanted to see, yes, nitric oxide is not killing everything, but is it because it's overwhelmed, or lower number of bacteria will be fine and we can fight it? So that was that work. Okay, I guess we can have a break now and then we can finish it. But please remind me, we should talk a little bit about tomorrow's test later in the class today. So, okay guys, thank you so much. Great. Okay guys, let's start our afternoon session. So now I can tell you that whole story that I was waiting for. Okay. Now, I showed you how NO releasing materials are great. They can kill bacteria in that CDC bioreactor. They can prevent clotting. I'll show you in future. One problem is they cannot prevent protein absorption. Okay, so if you have an implant, fibrinogen, protein, it's still sticking to the surface. So my lab over the years is working on lot of different anti-fouling strategies also. Okay, we think, so our goal is to go as close to endothelium as possible, the blood vessel that's carrying blood. Nitric oxide is only one of the mechanisms. We believe that in that final material that we want, nitric oxide will have one role. But we also know we need something more. Okay. And now we want, can we make a surface anti-fouling and nitric oxide releasing? So, if anything, protein, bacteria comes, this anti-fouling surface will prevent protein absorption, and nitric oxide will prevent platelet activation and bacterial contamination. Right? So, over the years, we have published a lot of papers that you can actually, if you are interested, message me, I can send them to you, where we have combined NO with slippery surface. That's what I'm going to talk today. NO with super hydrophobic surface, super hydrophilic surface, Zwitterion surface, or conjugation of bioactive agents such as heparin. All have nitric oxide, but we have tried different things with nitric oxide to see what is giving us the best synergy. Okay. So, I'll show you one example. Over the years, what happened? So, Marcus, my first PhD student, Joanna Eisenberg is a big name in our field, biomaterials and medical devices. Her group published a paper on slippery surfaces in 2011, 2012, around that time. It doesn't say. So the idea is you have silicon oil embedded into the walls of the tubing of medical devices, and tubing is slowly releasing that oil, and that oil is making the surface so slippery that anything like blood or bacteria or proteins are just slipping off the surface. They are not sticking to the surface. So surface is anti-fouling. So I talked that why can't we make nitric oxide releasing slippery surface? Because slippery surface is only preventing things to stick to it. They are not preventing platelet activation, and they are not killing bacteria. So Marcus was the first person who started doing this work, and his paper was published in Nature Scientific Reports. So basically, this got a little bit messed up. So this is a control silicon rubber. SR is silicon rubber. NO is nitric oxide releasing surface. Li is liquid infused surface. Linorel is liquid infused nitric oxide releasing surface. And all this fluorescently labeled protein is fibrinogen. So on a silicon rubber, lot of protein sticking. NO surface, protein sticking. Liquid infused surface, protein is not sticking. And linorel surface, protein is not sticking. So we made an anti-fouling NO release surface. We also did a 7-day bioreactor study where we showed how for Staph aureus, the combination of linorel and compared to only liquid infused or only NO was significantly better than by themselves. We did NO release, and we found out that our NO release is pretty consistent over 60-day period. Only NO or NO plus liquid infusion. So liquid infusion is not hurting NO release. Of course, that's what we want, right? But you can also imagine that how we would be thinking when we were designing this surface, right? The question is, do we make NO releasing surface first and then we add slipperiness to it, or do we make slippery surface first and then make it NO releasing? Right? So all this is more optimization. The beauty of slippery surface is you can just swell silicon oil into the walls, just like we can swell NO donor into the walls. We talked about how you can make a solution of NO donor, put your polymer in it overnight, it swells inside, and then you let the solvent come dry. Similarly, we can put it in silicon oil can go into the walls, and then it very slowly keeps replenishing on the surface, so your surface stays slippery. Okay.
So when graph is fluctuating,
Yes.
If we see the dynamics should be higher and then slowly decrease, but why these fluctuations?
So this fluctuation you are a little bit seeing, it's not too much. I mean, see, if you see y-axis, it's 0.4 to 0.8 or something like that. So it's not that fluctuation is, so we have 5 nitric oxide analyzers. It's instrument to instrument difference little bit, but it's not too much. Standard deviations are very tight, and it's almost the same range of NO release. So then what we did, we bought some urinary catheters from market that are used in patients, and we made them. So we bought silver urinary catheters, control urinary catheters, NO releasing urinary catheters that snap, and slippery surface with NO releasing urinary catheter. And when we did a 7-day bioreactor study, we saw that our catheter was almost 2-3 log reduction better then only silver or only snap. The combination was better than anything else. We also looked under SEM, and you can see all this is E. coli. They are like rod shaped, and our slippery surface was totally almost clean. If it was not slippery surface, what would have happened? If it was just NO releasing surface, second image is NO plus slippery. If it was just NO. How will we see dead cells? That's good, but with SEM, we cannot see dead cells. I mean, you might see a lot of bacteria on the surface, but if you do live dead staining, you would have seen a lot of dead cells. But here, you wouldn't even be able to tell that there are a lot of dead cells. You would see a lot of bacteria stuck to the surface, but you don't know anything else. Okay. Then, Megan finished her PhD 3 years ago. She did. So see, I'm showing you, we started working on this technology in 2018 or 1918, and how over the years we progressed with this particular technology. We got some clinical strains of different bacteria from CDC. Some were strains, functions, and I'm just showing one here, but she tested like 6-7 strains of bacteria. Because we had a grant from CDC, they wanted 30-day studies with all those strains from CDC for us to show that our material works. These were very difficult studies because it was a 30-day bioreactor. Bacteria was growing like crazy sometimes. So she had to repeat some studies because of contamination issue. But basically we showed that we were doing very well with our material. Okay. From yesterday's class, who can tell me why do we need rabbit model, thrombogenicity model? Why can't we just do in vitro studies? Why do we need animal studies?
That condition, property, temperature, pressure, and everything.
And
Easy to handle.
Easy to handle, inexpensive, and you cannot do more than 2-3 hour study, and we can do longer term study, right? So yes, we did this. Then my PhD student Divine, he graduated 2 years ago. He tested those materials in extracorporeal circulation model. Okay, we little bit talked about it yesterday. So what we do is we take this tubing and we coat it from tip to tip inside the tubing with our NO releasing material. Okay. For example, if we were using that diazonium dilate chemistry, we would have done something like this. We would have first coated it with our diazonium dilate material and then top coated it with just the polymer. Now we were doing slippery surface, so we swelled the tube with SNAP solution first, NO donor, and then we swelled it with the oil to make the surface slippery. I mean, we filled the solution inside and then locked it, and then overnight or whatever time, then we filled the oil inside. Oil and NO donors went into the walls. So actually I don't have a picture of the tubing. The tubing looks green after NO is loaded in it. And we load oil in it also. So now your surface is NO releasing and it's slippery.
or something?
No, because the powder, that anode donor is green color. Green in color. Okay. Let me go a little bit back for this study. So, few years ago in 2014, first we wanted to see, forget anode donor, we wanted to see what is the best polymer that is most hemocompatible by itself. So we did a 4-hour ECC study where we saw that E2AS polymer, that is a copolymer of silicon rubber and a polyurethane, was the best. The reason is, this is platelet count every hour. I told you in that ECC model, every hour we draw blood and we count platelets. So when we did every hour platelet count, we saw this E2AS material maintained the platelet count the best. Yes, it dropped from to 60% at 4 hours, the blue one. But it was better than other 3-4 polymers that we tested. Okay. Then I also told you in that ECC model that at the end of the 4-hour study, we look at the clot formation in the chamber. So we found out that clot formed on E2AS was the smallest out of the 4 polymers. Plus, 80% of the circuits survived in E2AS, whereas only 25 to 50% circuits survived in 4 hours. Meaning, I also told you yesterday, one of the things we do with this circuit is we measure the flow rate over 4-hour period, right? So we measure flow rate, we count platelets every hour, we look at the clot formation in the end, and we also do aggregometry every hour to make sure that platelets are aggregating. That I didn't show here, but for PVC, SP60D60, SG80A, almost 25 to 50% of the circuits survived, meaning 75 to 50% circuit clocked before 4 hours. So the flow rate actually dropped to 0 in 2 hours or 3 hours. Only E2AS had flow rate of above 10 or 20 or 40, whatever it was, I don't remember now, and was keep going for full 4 hours. And remember, there is no given, there is no material yet. This was just a study to understand what polymer is good by itself. And there was a paper we saw that said that actually E2AS polymer binds more to albumin than fibrinogen. And maybe that's the reason why it was superior in properties, because albumin pacificates the surface, because it does not have that RGD peptide that fibrinogen has, which platelets and bacteria like to stick to, right? Okay, now going back to that slippery NO surface, you know, something happened with all my figures. That silicon rubbers R went, that linorels L went. So I don't know, something happened, sorry. So what we did here, we tested silicon rubber tubing, just the tubing by itself, nitric oxide tubing, NO releasing, liquid infused tubing, and linorel or liquid infused nitric oxide releasing tubing in a 4-hour rabbit model. By itself, if you see this graph, silicon rubber tubing, only 65-60% of the tubing survived. Like by 2 hours, some of the loops actually clotted. The flow rate went to 0. But NO, liquid infused NO surface, they all survived full 4 hours at least. But then when we looked at the thrombus area, you can see how the thrombus area was. And when you see on the right side, you can see how there was so much thrombus in the top one, silicon rubber, and how NO, liquid infused, linorel, they all were nice. And liquid infused was maybe better than the other two. Okay. Then we wanted to test it in a longer term model. Our intravascular model, we little bit talked about it yesterday also, and also that how infection can happen in that area at the hub, clots can happen, infection can happen. And who can tell me what all we do in this model? Catheter model? No. What is the model? What do we look to the study? We talked yesterday.
We could see that contamination at the point of injection, at the point of toxic, and at the point of invasion into the blood vessel.
Okay. So I want to clarify one thing. We are not doing any lock solution. So lock solution is one strategy. Like people can fill this catheter with a lock, antimicrobial lock or heparin lock, and that's fighting infection. This model is we implant the catheter in the animal for whatever time. Best I think we have done is 1 month. Now, every day we can go to that hub and try to draw blood and infuse medicine or buffer or saline to make sure that the catheter is not clogged. Also, we can do is at the end of the study, we look at the clotting of the catheter with the imaging and SEM, and we can take pieces of the catheter to look at the infection of different places in the catheter. Okay. One question, how do you think we can take the catheter, how do you think we take the catheter out? So we implanted the catheter in the rabbit. After 1 month, I want to take the catheter out. How would we take it out? Surgery, sure. Then what will we do? Yeah, I mean, yes. So surgery, so yes, it's 1 month now. I want to take the catheter out from the rabbit. Can I just pull the catheter out? Okay, why no? So many people said no.
Catheter whatever is sticking on the surface of the catheter that might be disturbed.
Great.
So
Yes.
first, I think we need to sacrifice the animal, then take out the entire blood vessel that we have inserted our catheter in.
Very good.
And then in sterile condition, we
Yes. So what we do is we cut the blood vessel very nicely, and then we take the catheter out. Because if we pull it out, if there was a clot or whatever, it'll get stuck inside, and we might see very clean catheter, right? Okay. Ooh. See, this is what I was saying. I have some slides that are repeated. Okay. Now slide number 74. Great. Okay, so now we did this another interesting study. My student Elaine, I have her picture maybe later somewhere. She's defending her PhD on March 11th. So I reach March 9th and she's defending a day after. So she's very nervous. My flight should not get delayed and all that, you know. So she did this study. We went to a conference, and at a conference, I met somebody from FDA. They were mentioning about this study, which we found was very interesting. So what the study is, if you see that cartoon there, you on agar plate, you put a rabbit skin, and through the rabbit skin you put a catheter. now one side of the catheter you put some bacteria, and now you wait for 24 to 72 hours to see how much infection is going to the other side of the skin, mimicking our condition, right? If we have a catheter inserted in the body and some of the catheter is hanging out, we have this interface where skin catheter is going through the skin, and that's a very dangerous spot for bacteria to grow. So we wanted to mimic that in ex vivo skin study, where we have a catheter, here is a skin, you infect this side, and bacteria is traveling inside the body kind of through the skin. So if you see, the top one is control, and see how much bacteria is traveling on the other side. But this material, our material, is fighting and not letting the bacteria travel to the other side of the skin. Okay, so this was a nice, interesting study. Then I also talked that when we do the catheter study, we cannot just put the catheter in and do infection also, because if everything was done in a sterile environment, there is a good chance you won't see infection in a lot of catheters. I mean, chances of infection is, let's say, 5%, 10%, but then are you ready to do 100 catheters and hope 10 catheters will be infected and you can get your data? So better what we do is we actually infect the catheters before we implant them. Okay, so we dip the catheter for 2 hours in Staph aureus solution, and then we implant the catheters in the animal. Then we look after certain number of days how much is infection, because now we know all the catheters have infection. So we are actually making a very tough study for ourselves, that we are putting infection knowingly and seeing if our material can fight that infection. Okay, so now, and the beauty is we can get clotting data and we can get infection data, both. So we saw almost 70% reduction in clot formation, and see, that's the vessel we took the catheter out from. And for 28 days, so we took some catheters out at 14 days and took some catheters out. Some rabbits were 14 days, some rabbits were 28 days. And we saw 50% reduction in infection or clotting for 28-day data. Okay. But then, this was the really good data. I almost didn't believe in it, but we did N of 4 or N of 5. So there are 4 control catheters, 4 are lineural catheters. So basically what it's doing is, when the catheter was inserted, we took pieces from both ends of the catheter, the one that was near the skin and then one that was closer to the heart, and the tissue also, and we homogenized it and looked for infection. 14 days we saw almost no infection on one of the ends and some infection on proximal end and some infection in tissue. But for 28 days, almost no infection anywhere. So over 28-day period, our catheters fought that infection and just killed all the bacteria. So this was a very, very exciting study for us and very exciting data, that you are actually eliminating all the infection of a catheter. But, and you can see control had a lot of infection. So that means that infection worked. All the control catheters were infected. So infection is working just fine. And the standard deviation was so tight that it was done very, very reproducibly. So this was, and we have not published it yet. Hopefully we are going to submit it in next 1 month or so. Okay. So this is the story with this type of study. We have already talked about this. This video won't work, so I'll play a different video for you later. Whoa, oh, it's working. Wow, okay. Wait. Okay. I didn't know it'll not work. It'll work. Actually, it never works on my computer. Okay, so let's do one thing. This is an example of we developed a super hydrophobic coating. Okay, that means the contact angle is greater than 150 degrees. This paper is published. I don't know. I didn't put it here because I thought the video will not work, but I can send it to you guys. So what we did when we developed that coating, of course, we can coat tubes and catheters and everything, but we thought to make the study really tough, let's coat sponges, you know, and you can imagine sponges, like if you put liquid, it's going to just grab it, like, you know, and that can be blood or it can be bacteria. It's so absorbing. So here is a control. That's what you expect, right? A sponge should be. Now, this is our treated sponge with super hydrophobic material. Oh, what happened? Okay, one more time. So you can imagine if this sponge is, this is a coating, you are repelling everything, right? And here is another example. And imagine we did this study with some dye or something, but we can work with blood or whatever. This is a control, a sponge. Okay, and then okay. So the idea is how can we make the material so hydrophobic that it's repelling everything. Oops, sorry. Okay. Then now I have like some 20, 15 slides or something talking about different projects we have been working on. I wanted to show more, but I couldn't make more slides. The goal of my lab is more or less medical device, infection, clotting, in whatever applications we can go for. We have even published a paper on packaging materials, prevent infection in packaging, you know, just wherever we can apply that. You know, we have make sutures, fibers, hydrogels, coatings, films, tissue scaffolds, all these different applications. Okay, so here's an example. I have a good friend, Joe Potke. He works for US Department of Veteran Affairs. We are working together for 12 years. He's an expert in making microfluidic artificial lungs. Okay, so the idea is people waiting for lung transplant can have this artificial lung that they can carry, so that they don't have to be hospitalized and wait for a lung transplant. So here is an example of one of his microfluidic lungs. And you can imagine the size of the channels and small. So our role is how can we prevent clotting and infection of this lung? You know, the channels are so tiny, it's so easy for these channels to clot and then your lung will stop working if these channels start getting clotted, right? Again, just keep in mind the beauty is I cannot do the things that he can do. I cannot make this artificial lung, and he cannot do the things that I can do. So you have to learn to collaborate, and you have to rely on people from different backgrounds. Uh, Another idea, so I have this a friend and a collaborator, Dr. Andriy Baczynski. Um, he worked for Army. Now, last 2 years, he's not working for Army hospital anymore. But their idea is, if a soldier is badly injured on battlefield and they are badly bleeding, how can we keep them alive when their kidneys are failing, lungs are failing, heart is failing? How can we have extracorporeal devices that can keep that soldier alive for 2, 3, 4 days before they can be brought to a proper hospital setting? Okay, and you can imagine we cannot give heparin to this soldier because they are already bleeding, and if you give heparin, they'll just bleed much faster. So how can we make surfaces that will prevent clotting and infection but still keep the soldier alive? Another area of research that we are working on is instant clotting. So our company has a grant from US Army for this work. Instant clotting and bactericidal faster healing wound dressing. A soldier is badly injured in the battlefield and bleeding, actually bleeding so badly that they are not even concerned about infection. They are just concerned how to stop the bleeding so that they won't bleed to death. But at the same time, this type of environment in a battlefield, there is a lot of infection also. So how to take care of infection and prevent too much bleeding from happening with the wound dressing. So we are doing some work with the University of Miami, the company, and we are testing our wound dressings in a pig model there to see, can we clot, can we prevent infection of these wounds? Okay. Next, so these were my 3 PhD students, Megan, Jitin, and Arnab. And this is that NO generation technology coming back. I forgot to delete that slide earlier when I was saying. So we have published these different papers with selenium or copper in animal model. I think one of them, yes, this one was in animal model. The idea is we have a NO releasing polymer and we can put a NO generating catalyst on top so that the NO releasing polymer is sending NO donor, or sorry, not NO donor, only NO gas, and the top coat has the catalyst in it, selenium, copper, and when blood is coming in contact with that, it's generating NO. Now you are getting more nitric oxide release because you have a combination of NO release and NO generation both happening at the same time. The reason is, over the years while doing the studies, we also little bit found out, I'm not 100% sure, but we think the blood does not have enough packets of that RSNO, that S-nitrosothiol, that can get converted to NO when it comes in contact with the catalyst. You know, so if there are limited number of packets in blood that can get converted to NO, then NO generation can sound very fascinating because there is no reservoir. There is no limited amount of NO donor. You can have the catalyst and it can keep generating NO. We think the amount won't be enough either to have enough NO or to go for a very long time. So the idea is immediately after the surgery, when the chances of infection are high, you have NO donor and NO generation both working together. And now after some time, even if one of the technologies are not working completely, the other is still going. Okay. But for this type of work, now you can imagine, okay, if you have to design a coating that is NO release plus NO generation, what will you think about? Like, what are the factors you will have to keep in mind? Yes. Huh? Simultaneously. Yeah. Yeah, we want both of them to happen. Yeah.
Whereas we cannot coat our NO releasing material on the surface, it has to be inside.
Very good. Yes. The top coat cannot be releasing. Okay. And why?
Because the NO gel won't work. It
Won't work, right? Because it needs to touch that surface. Okay.
and the NO surface should be penetrative in it enough so that NO release is not hindered by that.
That mostly we don't worry. I mean, NO can easily escape. Yeah. What else?
So the top surface should not be reactive with RNA. Just act like a available surface just for the conduction of that reaction and not exactly bind with it in another fashion.
NO generating and NO releasing. Generating means it is.
Okay, I'll tell again. It's a little confusing. So NO releasing is you have a NO donor compound like SNAP, diazonium dialate that has NO bound to it and it's releasing it. We are putting that, we are synthesizing it and putting it in the polymer. That's NO releasing. NO generation is we are only putting catalyst, copper or selenium, let's say, and we are relying on the packets of RSNO that are floating in blood, that when they will come in contact with that catalyst, they will release NO that they were carrying in their packets. We are not putting a NO donor ourselves. We are relying on the NO donors packets present in blood. So it's just converting its packet to NO. So that's called NO generation. We call it NO generation. It's generating NO. We have published papers that are just NO generation also, where we didn't put any NO donor. Forget this green coating. We just put catalyst, put it in animal, and the blood in the animal is generating NO. But then we thought, let's go a step further and combine NO release and NO generation in the same coating. And that's what this was. But there is one more challenge here that you are not talking about. Who can tell me that?
Gel surface, the protein, whatever, that is going to bind on that surface. Bind. Because if a protein, if the surface is very clogged up with protein,
then we won't be able to do the generation. So very interesting. Wins. His paper is not published yet, but he has all the data now where he's making NO generating slippery surface. Now that slippery layer will prevent proteins to stick to the surface, so it can keep generating NO. Okay, that's a good thought. But there's another challenge in this that you guys are not talking.
SNAP needs to pass through that top material that gets
Yeah, I think he said or you said before, and that's not a worry. That's fine. We can easily, NO can easily pass. So why, oh, you said SNAP needs to pass. Why did you say SNAP needs to pass?
Because Snap is releasing that anode, but not Snap needs to pass, but the anode from Snap needs to be released.
Yes, and that's fine. That's easy. That's not a problem. Yes.
The combined release of NO-gen and NO releasing substance of the combined NO release should be optimal, that it doesn't induce any toxicity.
Sure. The levels should not be very high.
My doubt because like generation and that's why I asked you that both the levels should or it is releasing and generation the same process.
Oh, so NO release and generation does not need to be same. I mean, maybe initially NO release might be 3 flux, NO generation might be 1 flux. Over time, NO release might become 0.5 flux because NO is finishing, SNAP is finishing, and NO generation can still stay at 1 flux. The goal is their combination is good enough to do the job. So that's fine. But there is one thing that you guys have not thought. Take 1 minute to think about it.
Both the systems active together or in combination.
Oh, that's a good idea. Now, it can be very nice if we can on and off systems, but the problem is we cannot. As soon as I put it in the body in moisture and 37 degrees and all that stuff, how can I stop NO release not to happen? Or how can, see, if I can stop it, then I can be like, okay, for this time it can get on, this time it can get off. We can do that if it's external wound dressing. Then I can do is shine a light so you can have lot of NO release. Stop the light, your NO release is there but very low numbers.
So it will already happen. Like if during the day we are working, playing, or doing whatever, we're exposed to light. When we go to sleep, usually people sleep with dim or no light.
Yeah.
That can be conserved anyway.
Yeah, but now I'm just saying this is an implanted device. Yeah. So one thing that you guys are not thinking is, if you, so let me forget this picture. What will happen if I have a film that has NO releasing polymer, let's say SNAP, and in the same polymer I add copper. So I have copper or selenium plus SNAP in the same polymer. What will happen? Is going to? Okay, and then what will happen if they react? No. Why not a good thing? What do you think will happen?
That will be toxic.
No.
Snap to release anode because it is now binding or interacting with cellular copper.
No.
This is not the case. I think over the time when NO is being released from SNAP, some of the material might get loose in whatever some pores can be generated into that, and that can induce protein.
So the anodes themselves will start reacting with the copper selenium inside and not being.
No. So when I talked about that SNAP slide, I said the trigger to release NO is heat, light, and metal ions.
So it will start releasing a lot of.
So, see, SNAP is one type of RSNO. It's synthetic. We made it. RSNO in blood is also that family, but it's more endogenous. So, if we put SNAP and copper in the same coating, whatever that packet was reacting with copper, now that is, we are putting both of them together in a coating, they can start releasing lot of nitric oxide very fast. Because NO SNAP is breaking to release NO, but copper is causing it more catalytically release a lot more NO, and now more NO is releasing. Okay. It can be toxic or may not be toxic, but we might lose all the NO very fast because all the snap is getting consumed fast because of copper. So when we did this study, I don't remember, it has been few years, whatever, 2018, 2019, I don't know what years. We found out that NO levels were very high. So we put a sandwich layer between snap and copper so that they don't interact with each other. Now NO is coming out and copper is interacting with the packets in blood. It's not interacting with the snap in my coating. When we were not putting this sandwich layer, copper and snap were touching each other, and now we were releasing a lot of nitric oxide. Actually, it was so much that even while making the film and drying process, we were losing some of it just in the drying process.
What is the approximate thickness of each layer?
Ooh, I don't remember, but we can just make whatever. I mean, it's up to us, right? Like whatever the requirement is. So for catheters, if you want a catheter to be 2 millimeter diameter, outer diameter, we can have a total inner diameter of, let's say, 1 millimeter, outer diameter is 2 millimeter, so we can have that thick, that is our working thickness, right? Here, if we have a coating of a tubing, we can make 100 micron thick coating. It just depends on the application, how much room you have for thick or thin coating. But yes, thinner the coating, less snap you can put in. But the beauty of that swelling method, you remember snap swelling inside the tube is impregnation method. We don't need to coat it. We can just put all the NO donor in the wall of the existing tubing and dry it, and all the solvent is gone, but all the snap is trapped inside the thickness of the wall. So that's the beauty of that method, that you don't have to worry about thickness.
Something if we like any mesh-like surface, if we electroplate it with whatever desired material we have, okay, and then we coat it with something that is preventing the snap to react with the metal ions, so we can create a conjugate layer which will be even more thicker. I'm not sure that it
Okay.
will be thinner than this for now, but
Completely understand.
let's say you were talking about whatever a polymer, if we impregnate it with snap and everything. So we are just putting the snap in that. So what if we take the polymer, we electroplate it, like it would be a very thin layer of whatever metal that we are doing. We electroplate it, put a barrier layer like this in the mesh itself or in the polymer itself, and then impregnate it with snap. So this might reduce the thickness.
By reducing the thickness of the copper electroplated layer.
Maybe of the copper electroplated layer and the overall thickness of our material, whatever we are trying to.
But see, we have to also remember, if you are coating a tubing inside of a tubing, can you do this method inside the tubing? Right? Let's say you have a 1 meter long tubing and you have to coat inside of that with that method. Can we do all that? And how much will be the cost of that method? So, I mean, for a publication, maybe yes, great idea, we can try it. But can it become a practical solution? Those things will have to be figured out. Right?
One related to what this question was, have you ever studied to what extent that this are whatever NO generating material, what is the depletion rate of whatever metal that we are using?
Yeah, so usually it's not rate limit. Usually catalyst is not a problem for us. It will keep going. But do we have enough packets in blood to keep going? That's a more of a concern than our catalyst. Okay, we already talked about this. Then, this study is, so Akram was a postdoc in our lab and he did a lot of good work. So he did his PhD at University College London and he was very good at making super hydrophobic surfaces. So this work, like you can imagine, see the contact angle is 154 degrees on his surfaces. So this is a silicon rubber and after he adds his coating to it, it makes the surface super hydrophobic. And he also did is, he actually made this surface silver and nitric oxide releasing both. So you know silver is a standard used in industry right now. So we thought why not do a surface that's like combination of silver and nitric oxide and super hydrophobic. So it's super hydrophobic, silver releasing, NO releasing surface. And we did a lot of studies. It's a good journal. And we saw that combination was not cytotoxic. Last graph, we were able to reduce infection. Combination of everything, silver, NO, super hydrophobic, was better than anything by itself. And it also prevented clot formation on the surface because of super hydrophobic and nitric oxide. Not because of silver, because silver has nothing to do with clotting, but super hydrophobicity and NO prevented clot formation. Let me play some videos for you. So again, some of all these videos are Akram's work from different papers, some sponges, some tubings and all that. So video number 4. So let's look at it again. So polyurethane, these two, this is a control and all others are coatings of different type. And I think he had a water droplet, coffee droplet, and I don't know, he had different like Coke. Of course, we did it with blood also. So he had different types of solvents that he can repel a lot of different things. Okay. Now let's do number 13, the white, this one, the white sponge. Now this is just a sponge, a regular sponge, right? Then number 13, the gray sponge. Now it's a coated sponge with our material. Oh, see the video is so fast because the blood just goes.
0-second video.
See,
Less than that.
yeah, see. So, and imagine sponge is so difficult for things to just. Okay, now let's watch a tubing video. S8. Now this is a regular tubing in blood. We are just dipping it for whatever time and very fast blood is starting to stick on the surface, right? Now this is our coatings that we have put on. This is no nitric oxide. Just totally clean. Different types of tubings, different treatment. Equally, yeah. So they all are coatings that are, yeah. So the controls were this video. Some are slippery, some have super hydrophobicity. They are just different combination. This is,
We don't want to make the surface very hydrophobic, but so that it can absorb some amount of water. It was in the case of some other coating. For some type of coating, you said that we don't want the material to be very hydrophobic.
For endothelialization, we need something that control the
yeah, so sometimes, see, if you want things to grow on it and stick to it, and if it's so repelling and it's a permanent implant that's going to just stay there, now that can be an issue. Yeah. Okay, now video number 14. So again, same sponge with some blood study, right? This is all blood on it. This is treated sponge. Oh, something happened. It didn't work here. Okay. Yeah. So, okay. So these were some Akram's videos. Okay. We also talked about this work where we are trying different levels of bacteria, and with time how NO can fight to certain level very well, and then it still fights, but not at 100% or something. Okay, Vijay was a postdoc in our group, and he had a lot of experience with virus bacteriophage type of work. So this work was NO plus bacteriophage technology. That how we can act, NO is killing, bacteriophage is killing, and how their combination can be very effective. Okay. Then Dr. Chad Schmidt is my very good friend. Actually, last 5 days he was in India. He went to a conference in Jaipur, but we couldn't meet. I mean, he's in my university, so I see him all the time. But meeting in India would have been special.
That's why you said Jaipur temperature.
Yes. So, and Ryan did his PhD with me. So this paper was NO release plus heparin combination. Heparin is also a standard. So can we combine NO with heparin and show their synergistic effect? So that was the idea here. Lot of time we want to do this type of studies because convincing clinicians to just go away from the standard might be more difficult initially. But if we say, okay, you use silver catheters, that's great. How about NO plus silver is better than silver alone? Or how about NO plus heparin, it's better than heparin alone? So we did some rabbit studies with that. Then Arpita is a postdoc in our group. She did her PhD from IIT Guwahati. And this was her paper, making liquid-like omniphobic solid slippery surface, which is super anti-biofouling. You can even see on that image, there is a lot of biofilm growth on the top, Staph aureus and E. coli, and our surface had basically no biofilm growth. So this was that omniphobic anti-biofouling surface. Then this is another interesting work. We had a grant from NIH, we have a grant from NIH, and we have submitted a renewal. This work is with Dr. Chris Sidlicki, who's a professor at Penn State, and Elaine is a student who's defending on March 11th. So his technology is making these pillars on the surface inspired by shark skin, so that if bacteria wants to sit on the surface of these pillars, it's very difficult for bacteria to properly adhere to the surface. But then we went a step ahead and we thought, why don't we make these pillars NO-releasing? So we have these NO-releasing pillars. And in this work, we did some animal studies and we did in vitro studies. In vitro study was, we had these blood bags. His group did this work. We did animal work. And we had pump. We had catheter sitting in the blood. And then over time, we saw how textured really looked better than control. And NO-releasing textured looked even better than textured. And same for animal work, the combination of textured or NO-releasing pillars were much better than NO-release or pillars by themselves.
For animals being done, so is it
Approvals to do blood studies with animal blood is much easier. Human blood is also okay. It's just more process and more approvals needed. So it's a little bit more complicated than animal blood.
similar to animal model?
Yeah, something, because of course, whenever humans are like, it's more protected, right? Animals also, animal studies are more protected, but if you're drawing animal blood, that's little less harmful to animals. So that's why it's a little bit more easy. Okay. The next study, Priya did her PhD in our group 4 or 5 years ago, and Dr. Lachlan is my very good friend and collaborator. We have published 5, 6 papers together. So this work was NO-releasing zwitterionic surfaces. So zwitterions are hydrophilic surfaces, and you can see that, you might not see it very clearly because these catheters were so small. We measured the contact angle outside the catheter and inside the catheter with zwitterion. So the contact angle is much less, like maybe 40 degrees contact angle with zwitterion. And we did an animal study and we showed that how ZNO or zwitterion plus NO surface had much less clot formation compared to control. Like control looks terrible, right? And NO looks good, zwitterion looks good, but the combination looked even better. Then Megan did this paper on NO-releasing urinary catheters. So we bought urinary catheters, we made them NO-releasing by our method of swelling snap into the walls of the catheters, and did bacteria study, toxicity study, and everything. I'm just like, my goal is to just give you big picture. If something is really exciting to you, you can go find the paper, read more about it. And honestly, if the papers were published 4 years ago, I don't even remember the nitty-gritty details on the paper. Okay, this was also a good work. Mark did PhD, and now he's a faculty member a research faculty member in our department, NO releasing hydrogels. So these hydrogels were fluoride releasing also, and the goal was preventing infection and can we prevent demineralization of the enamel. So this study was very cool. We did a lot of bacteria study to show that with this was pluronic hydrogel, alginate hydrogel with fluoride ions in it. So what we did is we treated the hydroxyapatite discs with our material, and then we put demineralization solution in it, and the one that was not treated with our solution, this happened, all these pores started forming, and the one that was treated with our solution, it had strengthened the enamel so it does not get that demineralization happening on it. Next study, Morgan was also a PhD student. She did that catheter lock solution study. You lock the catheter with the solution. Now, what's the benefit of nitric oxide releasing lock solution? Yes, but most lock solutions are either heparin or antibiotic. They are either you're fighting clotting or you're fighting infection. Our lock solution is fighting clotting and infection both. So this was that type of study. Then Sama also did PhD. She worked a lot on silk. We published multiple papers on silk. So she made this gelatin silk scaffold, NO releasing. Basically, this was her gel. It helped in antimicrobial properties. I purposely am showing you different types of studies, that is like zone of inhibition study from those gels. And this gel was really good for tissue engineering type application because it helped in cells to adhere to the surface. And it made the gel, this network of silk made this gel very strong and not very fragile that will just break. So you made it stronger and antimicrobial and easy for cells to grow, so great for tissue engineering type applications. Dr. Melissa Reynolds, she works with nitric oxide work. I know her for 15 years now. We have published together a lot. We worked on this project, so she works a lot on MOF, metal organic frameworks. Idea is same of NO generation, but sometimes if you just add copper can leach out and cause toxicity. Metal organic framework, it's a bigger molecule and it's more secure and it does not come out as a leaching so easily while doing this NO generation thing. So that was this idea. Then almost done, few more slides, then we can talk about the exam. Nitric oxide is not very strong for antifungal applications. Okay, and you can see that in this graph, the bottom one at the corner. C. albicans, nitric oxide SNAP, it's little better, but it's not great. So this idea was, can we immobilize amphotericin B, which can take care of fungal infections, and nitric oxide can take care of bacterial infections. So now nitric oxide can prevent clotting, and nitric oxide can prevent gram-negative, gram-positive infections, and amphotericin B can take care of fungal infections. Arnab did this work. on antimicrobial peptides, contact killing, niacin. So if bacteria comes and touches the surface, it will kill it. So, and NO is always there to kill. So NO is killing things, but if somehow bacteria is still surviving and coming to the surface, niacin is killing it. Now, this was another cool technology, snapicillin. So ampicillin is a common antibiotic and SNAP is a NO donor. So we covalently bound SNAP to antibiotic. So ampicillin and SNAP, and we call it snapicillin. Now the idea is antibiotics cannot penetrate biofilms, right? You need 1,000 times the dose to penetrate. But NO can penetrate biofilms. So the idea is this snapicillin, if you have a surface that's badly contaminated, you expose it to snapicillin solution. NO will penetrate antibiotics and can kill the bacteria, disperse the biofilm. I don't know if I said the right thing. I just said it right. Okay. So you have a biofilm and NO will go inside the biofilm, disperse it, and now antibiotic and NO both can kill it. But by itself, antibiotic cannot kill a biofilm. And here is that data. So what we did is we grew MRSA on different surfaces, on a polymer surface, let's say. And then we exposed it to ampicillin solution. So first of all, we looked at MRSA and we homogenized it on a catheter and we saw this much bacteria was there. Other surface we exposed to ampicillin solution for whatever time, I don't remember. And then we homogenized it and we saw so much bacteria was there. We exposed it to SNAP solution, and when we exposed it to snapicillin solution, it took care of antibiotics better than anything else. So one paper is more solution paper, snapicillin. One paper is more polymer that has snapicillin releasing properties. Then, I think 2 more slides. I'm also getting tired. NO releasing anti-fouling surface. We did a animal model on it also, the ECC 4-hour model. And you can see the contact angle. When you had only NO surface, the contact angle was high. When you had the NO releasing anti-fouling surface, contact angle was much lower, and it worked great in animal model also. The last one is NO releasing sutures. So we made PLGA NO releasing sutures, which can now fight infection and reduce infection, and over time the suture will go away anyways. So that's the idea with NO releasing. And Lauren did her PhD last year. So, and she's working for the company now. So to conclude, we did a lot of things today, but main thing is we can release nitric oxide for long periods of time, right? That was one of the challenge when I started working in the field. We can swell or impregnate SNAP into the walls of the materials, so we don't have to extrude. So that was another challenge. We can sterilize it. It is shelf stable, which was another challenge. NO releasing materials do not prevent protein absorption. So we can combine it with super hydrophobic material, super hydrophilic material, slippery material. So we can take care of that NO plus anti-fouling challenges. And we are working on catheters, stents, grafts, urinary, wound dressing, endotracheal, dialysis. This is like so applicable to so many different applications. So really cool technology can be very easily combined with antibiotics, heparin, hydrophilic, hydrophobic, whatever type of other technologies you want to combine it with. It's very easy. You can make hydrogels, or you can make sutures, or you can make films, or so very versatile technology. And with that, I want to thank a lot of people, a lot of my group members who did a lot of this work. I think in last 10 years, I have not done a single experiment by myself. So of course, the credit goes to all the students who worked in the lab. I had so much more data by other people. I just ran out of time. So if my students are upset that I didn't show their data, I'm sorry. And of course, thankful to so many collaborators. They all have so much different skills that we don't have. Funding agencies, if you don't have funds, you cannot do anything. So yeah, so this is what approximately what our lab does. Question. Okay, great. I mean, you didn't clap for me for 7 days and today you want to do that. Okay. Okay. So, exam tomorrow. Let's talk about that. So, any questions for exams? Yeah, for exam. So, exam will be 11 to 1 tomorrow. And my understanding is if you don't take the exam, you don't get something. You still get your certificate, but you don't get your credit. It's a 2-credit course or 1-credit course, whatever it is. You won't get that credit if you, so you won't get your credit if you don't do the exam, right? First, for online reasons, let's little bit talk about big picture of the work I talked without talking nitty gritty details. Do you have any questions? Yeah, but I mean, if you have some other questions or any ideas or anything, what you think?
Told one of your collaborators the star felt like
Yeah.
inside the building. Use it inside the building.
So the work there, that's interesting question actually. It's interesting question. So what they do is they make that sheet and I think they roll it into a catheter. Uh, Making it inside a small catheter is very challenging. That was your question, right? How they make?
Was it pre-manufactured or did you fabricate it?
No, so I think they make it into a film and then they roll it into a catheter, because going inside a 2 millimeter diameter catheter and making those is, yeah, how do you do that? So see, that's a good example of how do we do commercialization with that type of material, right? So that's a little challenge that we might face with that type of technology.
But if you were fabricating, you could have casted it on something like a pole or anything that has all these grooves and you.
Yeah, but see, I don't remember exactly, but I think those pillars are half a micron apart. You know, I think so, because if the size of a bacteria is 1 to 2 micron, those pillars are maybe 700 nanometer or something like that. So they are like so close, making it in a tiny lumen of a catheter can be really challenging. Okay, so if I ask you, out of all the stuff I showed you, okay, no, you all have answered separately. All the different types of things I showed you, what was most exciting for each one of you? Like, you know, I showed so many different papers. This is Dysanium dilate, SNAP, Zwitterion plus NO, this, that, suture. What technology was
It was really nice. It was really innovative. use two materials, like one which was generating NO and one which was
So NO release plus NO generation was you liked a lot.
I got something in my mind which I'd like to share.
Okay, okay,
But yes, it can be something.
okay,
It is
okay, okay.
I love Snappy one because the next pandemic. And so, as I don't know about USA, but in India, antibiotics are so available over the counter, and we're just taking it like candies.
Yeah,
So, and we're not even completing the dosage.
yeah.
So, that could be the next thing. And we can obviously foresee just like pandemic, like COVID, or the antimicrobial pandemic that's ahead. So we need some technology to curb it. So I really love the idea and the idea that NO being a gas can penetrate that wall, whereas 1,000 times more of the antibiotic dosage could not pass the filter. It's
Yes.
very efficient, very doable, and I believe when the need comes, it could be scaled up to a larger extent.
Okay. So one person liked Norel plus Nogen, one person liked Snapicillin. We have a grant from NIH for Snapicillin, and actually we have not published and we have not filed for patents, but we are making different combinations.
Yes.
So we are making that similar style NO antibiotic, different types of those. Okay. Do you have any that you particularly liked? Hydrophobic. Yes. Okay. Anybody? See, everybody does not need to answer, but if you feel something you really liked, then you can tell me. Either you didn't like anything or you liked everything, and that's fine. Anything that jumped out? You mean where we put snap into the walls of the tubing? Okay. So diffusion method to impregnate the tubing. So see, that's a very practical, I mean, all of what you are saying is useful, right? I mean, I did it, so I think it's useful. That's fine. But see, Snapicillin has one type of advantage. Norel Nogen is different. But yes, you have both technologies working together. Your style, what you are talking about is it's very helpful for manufacturing side of things. It makes it easy to manufacture. Another good one that I really like is this Linorel, liquid infused nitric oxide releasing surface, and we have trademarked that name also for our catheter technology because you're making the surface anti-fouling and antimicrobial and hemocompatible, everything with that technology. And that actually the animal data showing all the bacteria dying on the catheter, I really, that's like, I like that a lot actually.
There was one thing like artificial skin.
Yeah.
That is also very interesting, and I think
So the thing is, we have not pursued too much of that. You know, lot of times this research is very grants driven also. The area where we have got grants or the area we feel we can get future grants, we lot of times want to go in those directions. No, I know. I like it too. But another thing is, if you don't have funds, doing animal studies is very difficult because you cannot use one grant money to do a different animal, right? So a lot of times students who want to do studies, they want to do catheter or ECC type work because we have funds to do those type of animals. But if a student develops a really nice artificial skin, we don't have funds to test it on animals. We can write proposals, but that is our 2-3 year project. And if you're lucky, you might get funding. So I would love to do that more, but so far we have not been able to.
For some that for a burned skin that it was I guess hypothetically with someone they used some skin of a fish to make the wound so that the skin can do again or something like that. So it was very interesting and then I saw the
Yeah.
artificial skin today so I thought that somehow like you can use some animal skin on humans to
Yeah.
treat the wound or how
I know.
how does that work?
No, I mean, I don't do much of that work. We are doing wound healing with U.S. Army project, but not that artificial skin too much. Anything else? Okay, so we can talk about the exam quickly. I have the paper ready. I think it will be a 2-hour exam. And 15 multiple choice questions, 5 short answer questions, and 2 numerical problems. We talked about polymers, ceramics. We talked about protein adsorption, platelet activation, bacterial infections. We talked about 3 different types of metals: stress strain, molecular weight of polymers, weight average, number average, and that bacteria, like the generation time of bacteria. It will be a closed book exam, so you need to remember all the formula, and no cheat sheet or anything for formula. You need to remember those. Actually, yeah. So it's just, yeah, 50. Yeah, doesn't, yeah. So
For numerical type question, do you expect only answers or do you expect the steps?
I would like steps, because when you are sitting next to each other, writing an answer is much easier. If your answer is wrong, you will get a 0. But if you show steps, I will give you some points for steps also. What if you made a mistake in the last step? Then you might only get 10% points deducted. So always show steps. Calculator is allowed. Phones are not allowed, whatever. Yeah, I mean, yes, because you see, our work is such that you can easily Google stuff, right? So, yeah. Yes.
Actually, the question I want to ask is from some days before.
Oh.
Coagulation cascade you talked about. I tried to understand it, but I just want you to summarize in a way that.
So I'm trying to understand it for 10 years, and even I have not understood it. So you're okay.
In tomorrow's exam, to ask any question from that, I actually have to understand some part of that.
Okay. So let's talk about it a little bit. So there is extrinsic and intrinsic pathway for coagulation, right? And you can remember, extrinsic is injury. So if there is an injury, platelets are important to immediately plug that hole. If it's an intrinsic pathway, protein adsorption happens first, and then platelets are sticking to it, right? Both pathways lead to a common pathway where factor 10 gets converted to activated factor 10, which helps convert fibrinogen to fibrin, which results in cross-linking of fibrin and forming a mesh.
Which started starts this cascade.
So even if you don't, I'm not going to ask you that cascade. I can tell you it. But there is a factor 11 that gets activated on one side, factor 7 gets activated on other side. So those are the factors that get activated.
They're only explain.
Uh,
Injury side.
injury side is tissue factor 7. Yes. So they start and then they come to common pathway. Common pathway releases chemicals that keep generating more and more, creating more factors, right? Another thing you should know is how injury gets plugged. So if somebody gets a cut, collagen gets exposed, they recognize that collagen, they stick to that collagen, WF, and then they activate and release thromboxane or TP, activate there, and they activate and bind to each other. Fibrinogen comes, acts as a glue between platelets. Fibrin forms, cross links, results in a clot formation. That's how bleeding stops. Or I think if you know this much, you're fine. I'm not going to ask you the whole. If you tell me to draw a whole coagulation cascade, I cannot either, right? Right. Uh, so this much is good if you know. Okay, guys. So thank you. I'll still be here. We can talk, but session is good, and we'll see you tomorrow for exam at 11 o'clock, and then we will do some class at 2 o'clock tomorrow. So, okay. Thank you.